we'll try to understand how the pattern of acetylation influences the biological activities of partially acetylated chitosans. So this being a seminar for the European Chitin Society, of course, I don't have to introduce chitin or chitosan. But what I want to show with this first slide is that we all know that chitosan is chitosan is the most fascinating and most promising functional biopolymer, but we also know that it's not always fulfilling these promises. We often have problems with reproducibility, especially when it comes to biological activities. So for instance, we see here the agricultural applications of chitosan, where chitosan can protect plants from disease, or we see wound healing properties of chitosans. But we know that it sometimes works and it sometimes doesn't work. And so already 20 years ago, we decided that we have to understand structure-function relationships really in molecular detail, so that we know which chitosan is responsible for which bioactivity. And then it should be possible to have a chitosan which reliably and reproducibly always gives the same bioactivity. And this, of course, would be the basis for the development of chitosan-based applications. So as Gregor already mentioned, we started initially to look at the degree of the influence of the degree of polymerization and the degree of acetylation on biological activities. And to visualize these, we came up with the chitosan-matrix, in which we have on the x-axis, I can try to use my pointer. On the x-axis, we have the fraction of acetylation from fully deacetylated to fully acetylated. And on the y-axis, we have the degree of polymerization from the monomer to the oligomers, to large oligomers, small polymers, and then to real large polymers. Now, of course, on the upper right-hand corner, we have the insolubility range. That's the range where we have chitin, high degree of polymerization, high fraction of acetylation. So this is crystalline chitin. That's not so easily used for biotechnology, but all the rest of this matrix is available. And then we teamed up with chemists, with Alain Domar and Kjell Vorum, both of them past presidents of our European Chitin Society. And they developed for us series of chitosans which differ in one parameter only. So for instance, we have a chitosan with an intermediate low molecular weight polymer, and then this was fully deacetylated in the beginning, and then chemically we have N-acetylated it to give a series of chitosans with the same degree of polymerization but different fractions of acetylation. And in the same way, we had a large polymer which we partially degraded to get a series of chitosans which have the same fraction of acetylation, but which differ in their pattern, in their degree of polymerization. And using these chitosans then, which only differ in a single parameter, we started doing bioactivity assays. So as plant biologists, and plant pathologists, we were mostly interested in plant strengthening activities on the one hand and antimicrobial activities on the other hand. So for plant strengthening activities, we performed many, many different assays, and I'm only showing one here in which we injected chitosan solutions into the intercellular space of wheat leaves. And then 2 days later, we can see this reaction here. And here we injected a series of chitosans with a fixed degree of polymerization, but different fractions of acetylation. And you can very clearly see that a fully deacetylated chitosan is completely inactive. But when we increase the fraction of acetylation, it becomes very active. But if we increase the fraction of acetylation even further, then it becomes inactive again. And at Fa 0.6 or 0.7, so close to chitin, it becomes completely inactive. This was an extremely important early result we had, showing that it really matters which chitosan to take. So if you just take any chitosan from the shelf without knowing what is its degree of polymerization or what is its fraction of acetylation, then you cannot expect it to have reproducible activities. You have to take the right chitosan if you want to have a certain biological activity. The same, what I'm showing here for a fraction of acetylation is true for to somewhat less extent for the degree of polymerization. So it really is a shame that even today, we recently calculated about 50% of the papers appearing on bioactivities of chitosans do not report the degree of polymerization or molecular weight and the fraction of acetylation of the chitosan used. So in that case, these papers are more or less useless, because if you don't know which chitosan was used, you cannot really reproduce it. What we found for plant strengthening activities was also true for antimicrobial activities. They also depend strongly on the structural aspects of the chitosan you use. So we found that the highest antimicrobial activities is shown by chitosans with a low fraction of acetylation and an intermediate degree of polymerization. Small polymers with low acetylation, low degrees of acetylation are best. Whereas for plant strengthening activities, we found that large polymers with medium or intermediate fractions of acetylation were best. So not only do different chitosans have different bioactivities are also carried by different chitosans. So if we want to develop a chitosan that can protect plants from disease, we need a mixture of antimicrobial activities and plant strengthening activities. So these chitosans, which are well defined in terms of their degree of polymerization and their fraction of acetylation, they are today called the second generation of chitosans, and they are now becoming available on the market on industrial scale, so that they are available for the development of applications. And this is why you see increasingly now you can see chitosan-based applications, chitosan-based products on the market. But this, as Gregor already mentioned, degree of polymerization and fraction of acetylation are only two of the three main structural parameters that determine the structure of chitosan. And the third one, of course, is the pattern of acetylation. So the distribution of the acetyl groups along the polymer chain. It took us a long time to start working on the pattern of acetylation, because the chemists told us it is impossible to generate chitosans with different patterns of acetylation. If you do your chemical process nicely, then you end up with chitosans which have reproducibly random patterns of acetylation. It's not bad, it's reproducible, you know what you have, but you know that you have random patterns. So it's not possible to find out what the influence of the pattern of acetylation would be on bioactivities. So we decided to try and find out whether chitin and chitosan modifying enzymes, such as chitin deacetylases, could be used to generate chitosans with non-random patterns of acetylation. So in order to open up the third dimension, pattern of acetylation, which can be random, that's where the chemically produced chitosans are, but in theory at least, it could also be blockwise, or it could be more regular than random. So we started working on oligomers because, of course, with oligomers, it's easier to control and also to determine the pattern of acetylation. We developed methods, mass spectrometric methods to analyze, to sequence chitosan oligomers. And then we also started cloning chitin deacetylases. And we found that chitin deacetylases are regio-selective. So they typically act on certain units within the chitosan and not on others. So for instance, here we have two bacterial ones, not B from rhizobacteria and VC-CDA from Vibrio. And you can see that they act very specifically. Not B only deacetylates the unit at the non-reducing end, whereas the Vibrio enzyme deacetylates the unit next to the non-reducing end, only this one unit. And the nice thing is that these enzymes can also be combined. They can act on the subsets of each other. So by combining them, we can have a chitosan, which in this example here, chitosan hexamer with two deacetylated units, and they are specifically the two units close next to the non-reducing end. We also did the same for fungal enzymes, and we found that fungal enzymes typically have a different way of deacetylating. They can typically deacetylate most units of a chitosan, except for some specific units which they cannot attack. So for instance, the one from Vaccinia graminis can deacetylate all the units except for the two units close to the non-reducing end, whereas the PES-CDA from Pestaloceopsis can also not deacetylate these two units, but it can also not act on the reducing end. And again, we can combine these enzymes, for instance, with the bacterial enzymes, and create even more patterns. So surprisingly, with just a handful of, or maybe two handfuls of chitin deacetylases, we were able to prepare all 14 possible tetrameric chitosan oligosaccharides. We can prepare them, of course, only on small scale, on milligram scale, but that's enough for bioactivities. And I will give you one example here. That's on the mono-acetylated tetramers. So the one that have a single acetylated unit, which can be at the non-reducing end, next to the non-reducing end, next to the reducing end, or at the reducing end. And these mono-acetylated tetramers we used in a priming assay, in which we have plant cells which are first treated with a priming agent. And these were our chitosan oligosaccharides. And then after a while, after 3 hours, for instance, that depends on the species you use, we treat it with an elicitor that induces an oxidative burst, so the rapid and transient production of hydrogen peroxide. And we use this elicitor at a low concentration so that the elicitor alone is inducing almost no oxidative burst. But if we use the elicitor on a primed cell, then we can see that the same low concentration of elicitor can induce a very strong oxidative burst, because the priming agent has sensitized the cell so that it can see this low concentration of the elicitor more readily and can quickly and strongly react. Now, when we use our monoacetylated tetramers as priming agents, then we find that the priming activity is very strongly dependent on the position of the acetyl group. If the acetyl group is at the non-reducing end, we have a very strong priming activity. The more the acetylated unit moves towards the reducing end, the lower the priming activity. Interestingly also, this one is a mixture of the two most active ones. So the one with the acetylated unit at the non-reducing end, and the one with the acetylated unit next to the non-reducing end. These two oligomers are in a mixture of 1-to-1. So what you would expect would be an intermediate activity between these two. But what we find is an even stronger activity. So it looks like these two monoacetylated tetramers have a synergistic activity. That's something which we repeatedly find now, that if you combine different chitosans, we can see synergistic activities. So this was an example on the tetramers. But of course, the real challenge is to try and generate chitosan polymers with non-random patterns of acetylation. And the problem here is that when we use these enzymes on chitin, on fully acetylated crystalline chitin, they are almost inactive. So it is not really possible to use chitin deacetylases to generate chitosans from chitin. So what we have to do is we have to do a first step of alkaline of chemical deacetylation to generate a chitosan with a high fraction of acetylation. Here is one of FA0.6. This can be produced either by partial deacetylation of chitin or by partial acetylation of polyglucosamine. And then this polymer, which has a random pattern of acetylation because it's produced chemically, can then be used as a substrate for chitin deacetylases. So when we do this using three different enzymes, three different recombinant fungal chitin deacetylases, we start with a fraction of acetylation of 0.6, and we use these enzymes until they do not act, until they come to the end of their activity. They do not continue further. And typically these enzymes stop at a fraction of acetylation of 0.3. So they cannot, all these three enzymes which we used here, they can reach DA30 or FA0.3, but they cannot further deacetylate these. And then we developed enzymatic mass spectrometric fingerprinting analyses to analyze these chitosans, and we found that they differ in their pattern of acetylation. So enzymatic mass spectrometric fingerprinting means that we use an enzyme with a known cleavage specificity, which cleaves these polymers at specific sites, and then we use mass spectrometry to quantitatively sequence the oligomers produced. And then we use principal component analysis, for instance, to analyze our results. And then we can clearly see that chemically produced chitosans are in a certain area here. So that we know this is random pattern of acetylation. And these three chitosans, which are produced using enzymes, or it's a chemoenzymatic process, because the first step is a chemical process and the second step is the enzymatic process. if we use the pestalocyopsis enzyme, we get a chitosan, which has almost random pattern of acetylation, at least in this PC2. It is where the chemically produced random PA chitosans are, but the PCT-CDA has a more blockwise pattern of acetylation, whereas the PA CDA produces a chitosan with a more even distribution of the acetyl groups. So using enzymes, it is possible to generate chitosans with non-random patterns of acetylation. So now if we go back to our matrix, we now have the third dimension, the pattern of acetylation in our matrix, and we have generated 3 chitosans which differ in their pattern of acetylation. One of them is close to random, one is more blockwise, and one is more regular. But what we can also see is that of the possible space, we have only covered a small part. And that is because the substrate we used, the DA60 chitosan, already has a random pattern. The starting material has a random pattern. So we cannot expect a strong blockwise pattern of the strong blockwise distribution of the acetyl groups after the enzymatic treatment. Now we have come up with a different way, which I'm not going to describe in detail because it's not yet published, but it allows us to really access the full range of this pattern of acetylation. So we can produce a chitosan with a very blockwise distribution, and this one is one with a medium fraction of acetylation, but we can also produce one with a low fraction of acetylation, and still a very strong blockwise distribution of the acetyl groups. Now, of course, the crucial question which really interests us is, does this pattern of acetylation influence the biologic activities? And I'm only going to show one example here, and now it's more a medically relevant example, and that is the transfection of human cells by chitosan nanoparticles, which include RNA for a green fluorescent protein. And what you can see here, this is the positive control, lipofectamine. So this negative control, of course, no fluorescence, this is the positive control. And you can see that this chitosan here, the low fraction of acetylation, but blockwise pattern of acetylation is as good, is actually even a little bit better than the lipofectamine. This is what you can see here. It's very strong, very strongly able to transfect the cells. So this chitosan, which has a low fraction of acetylation, actually this has a fraction of acetylation of about 0.1, so degree of acetylation 10%, but it has a blockwise distribution of these few acetyl residues, and this is very good for transfecting cells. And the interesting thing is, if you digest this chitosan with different enzymes, and there are also 2 human enzymes here, then you can see that this, even though it has a very low fraction of acetylation, it can be degraded by these chitinases, and because oligomers are produced, whereas a chemically produced random PA chitosan with the same low fraction of acetylation will not give you products. Especially lysozyme is interesting because lysozyme requires 3 consecutive acetylated residues for a single cleavage. Now, if you have a chitosan with a fraction of acetylation of 0.1, it means that 3 acetylated units next to each other is not very frequent. In fact, if you think about it, you really need 7 acetylated units next to each other to produce this A4, the tetramer. This is what we see here. We see a fully acetylated tetramer. To obtain this tetramer, you need 7 acetylated units in a row. So this is very unusual for a chitosan with a fraction of acetylation of 0.1. But our enzymatically produced chitosan has this acetylated block, which allows it to be cleaved by human enzymes. So of course it means that these chitosans can be used for biomedical purposes. So far, the biggest hurdle for internal use of chitosans in biomedicine is that they are not degraded. They are taken up by human cells, but they are not degraded inside the cells. But our new biotech chitosans, which have a blockwise distribution of the acetyl groups, they can be degraded by human enzymes. So we think this is a breakthrough for the use of chitosans for internal medicine. And of course, one example would be RNA-based vaccines, which you could use chitosan very nicely for transfection to bring the vaccines into the cells. But with our conventional chitosans, this is not possible because these chitosans would not be degraded inside the cells. But our new block PA chitosan will be degraded. So we are really proud of this, and we think that this really will lead to a completely new field of possible applications for chitosans on the biomedical side. So with this, I come to an end of my talk. I'd like to thank, of course, all my group, because none of what I've described here I've done myself. I'm only talking about this. The real work is done by my excellent students, to whom I say thank you. And of course, I say thank you to all of you. And I hope that you stay now, of course, for the second talk, and then to see you again next month, because this webinar is supposed to be a regular event happening every last Tuesday in a month. Thank you.
Thank you so much, Bruno, for this incredibly exciting talk about pattern of acetylation and how far you got also with the polysaccharide and polysaccharide level. It's really intriguing. So if there are questions, please unmute yourself and ask right away. There is also the option to write a question in the chat if you're not comfortable like verbalizing it. So please go ahead. No questions so far?
Well,
oh, yeah.
Oh, it's Mats Andersson here. Thank you very much, Bruno, for this interesting presentation.
Hi, Mats.
I just want to check that I understood correctly. Did you say that the random type chitosan doesn't degrade is not degraded by lysozyme in vivo? No, you didn't. I hope.
I did not get the question completely. I said that a conventional chitosan with a random pattern of acetylation, which has a very low fraction of acetylation of DA 10% or FA 0.1, is not degraded by lysozyme.
Oh, okay. No, then I misunderstood, because as you probably know very well, the 50% deacetylated or acetylated chitosans degrade very rapidly.
Yes, of course.
Yes, okay. Thank you very much for this clarification.
Thank you.
So maybe a question from my side before other question might arise. It's really intriguing, like this enzymatic approach, and I'm well aware about your first works on the kind of oligosaccharides, and now you could like bring it to the polysaccharide level. Just thinking about a commercial exploitation of this approach to actually access these polysaccharides. So upscaling, I think if you want to apply enzymes, either there's the whole cell approach or you immobilize the enzymes. So which potential do you think in that regard? Is it upscalable? Do you think that chitosan polysaccharides with defined patterns of acetylation can be produced like in a commercial scale with these enzymatic approaches?
That's certainly a very important question. What we can do in the lab is, of course, we can produce milligrams, which is enough for many bioassays, because, as you know, micrograms are typically enough to induce bioactivities. But for applications, more would be needed. We are just trying to set up a pipeline which would allow us to produce gram amounts. I'm very optimistic that this will work, and if this is successful, then I think kgs will also become possible. Beyond kg, it will certainly become difficult, and of course it will become expensive. Yeah. So such a chitosan I would not try to use for wastewater purification or for agricultural applications. But if you think about cosmetic applications or biomedical applications, then a kg can already go a long way. And then, of course, the price is high enough that biotechnological production would be possible. It's still a far away, but I think it's possible.
Okay, thank you. Vincent, there's a question from your side.
Yeah, I just wanted to thank you, Bruno. Very nice presentation, fantastic work. I was just wondering, I heard you say something that I really like to hear, is that you said it's basically not possible to produce chitosan from chitin by using deacetylases. I just want to draw everybody's attention to this remark. This is something that we also have been claiming, but nevertheless, you see these sentences in very many reviews about chitin processing, that we can make chitosan with deacetylases. Well, everybody, you cannot. It's impossible. And it's very obvious, I think, why it's not possible. And Bruno, I really want to ask you, do you really mean it? Because I do.
Well, of course, we know that in principle it is possible. Now, I think the problem is that the substrate is crystalline, and so the enzymes will probably work on the surface of the crystals.
Exactly. Yeah.
So that's why we get like 5% deacetylation. If we have an enzyme that's very good, we can reach 10%, but that's it.
Yeah, we see the same things. Yeah.
Yes. I mean, this has been shown by many people, but it's also possible if we break up the crystal, if we manage to get what is called nanocrystals or very finely dispersed chitosans, then it has been possible to use enzymes to completely deacetylate chitin.
Yeah, okay. Yeah.
In principle, it is possible, but of course, it's extremely tedious to do so. But I'm glad you say that that you like the comment, because I mean, it's only one of the many myths which surround chitosan. There are so many fairy tales. Martin Peters called it fairy tales about chitosans. We are just writing a review article in which we are trying to look at all these fiction and at these myths and try to sort facts from fiction, because I think it is very important. There's so many things said about chitin and chitosan and their enzymes, which are simply not true. But still enough is true to make it a fascinating molecule.
Absolutely. Thank you, Bruno. Yeah.
Yeah, great conversation, and I hope we can also contribute, like demystifying those fairy tales with this webinar series.
Yes.
Rupert, you had a question.
Yeah, thanks a lot, Mr. President, for this very nice talk. I just would have one regarding the conformation of this oligomer. So if you have the acetylation at different positions, how dynamic is the 3D structure of such an oligomer in solution? Is this known, how they look like, how they form and how they inhibit maybe or interact with enzymes then? Or are they very dynamic? So are they moving a lot in terms of conformation?
Yes, they are moving. We're actually doing quite a lot of molecular dynamics now to try and understand how they move in solution, and then how they interact with enzymes. That's what our interest, because then if we understand exactly